Hello everyone. I hope you're doing well and doing your work on mitosis. Today what we're going to concentrate on is the next part of chapter 5, which has to do with meiosis. And so we're going to move into sexual reproduction. But before we get into sexual reproduction and the concepts that are presented in this next few sections in chapter 5, I just want to make sure that some of the terminology is you're feeling comfortable with it, and so that I can use it and you don't feel like you're getting lost in it. It's pretty easy to feel that way when it comes to doing this genetics. So in body cells, which are called somatic cells. So when we're talking about our body cells, the scientific term for a body cell is a somatic cell. And in our body cells, or somatic cells, our chromosomes come in pairs. You've received a chromosome number 1 from your mom and a number 1 from your dad. They make up what is called a homologous pair. And you can tell the members that make up a homologous pair because they're the same size, they're the same shape, and they contain the same type of genetic information. In other words, if on chromosome number 1 is the information for hair color, it's on everybody's number 1. So hair color would be on your mom's number 1, it would be on your dad's number 1. Now it's possible that your parents gave you different forms of hair color. Perhaps you got the brown form from your mom and a black form from your dad. Those different forms are called alleles. So when it comes to doing genetics, we often speak of alleles. And those are just the different forms of a gene. So the gene is for the trait hair color, and there's multiple alleles out there for hair color. There's the blonde allele, there's a red allele, there's a brown allele, there's a black allele. So that's kind of some of the basic terminology. You will know the members that make up a homologous pair because they're the same size. And that's how we lay chromosomes out. And I'm just going to jump forward to a couple slides so I can show you a karyotype so that you can see that real images of them. Here's a real image of them. You can see chromosome number 1 is larger than pair number 2. 3 is a little bit smaller. So I think I said this in the last lecture, that they go in a descending order according to their size. They also have a different shape. And what's determining that they are a different shape is the placement of the centromere. So remember, the centromere is where the sister chromatids are still held together. And as you can see in chromosome number 1, the centromere appears to be pretty much right in the middle of the chromosome. However, if you compare that to chromosome number 4, the centromere is much closer to the top as it is in number 5, and that gives that chromosome, even though I know these are sister chromatids, but it gives this a different shape than, say, chromosome number 5. So if you were trying to find the pairs of chromosomes, it's pretty easy. This is how it's done. It's done through a photographic image where they've applied stains to the chromosome, and then they take a picture of them, and then they cut the photograph apart, and they start lining up and pairing up the homologous pairs. So you know, again, who makes up a homologous pair of chromosomes because they are the same size, they're the same shape from that centromere, and they contain the same type of genetic information. That would be the traits: hair color, eye color, arms, that sort of thing. Okay, so diploid versus haploid. We often speak of their ploidy level, and so a cell that contains both members of a homologous pair, such as your body cells. Those are called diploid, and the shorthand for that is 2N. So we refer to them as being a diploid cell, and I'm describing its ploidy level, diploid. So that's where that terminology comes from. The shorthand for it is to say 2N. Okay, so all of your body cells are 2N. They're all diploid, and that's what mitosis does. So the last stuff you were working on mitosis is you start with a diploid cell and you finish with 2 diploid cells. So mitosis completely conserves the ploidy level of the cell. It's simply cloning one cell after another. A cell that contains one member of a homologous pair of chromosomes is called haploid. Haploid cells, either they only have one number 1 in them, a number 2, one number 3, one number 4. So they have all 23 chromosomes in there, but they do not come in pairs. So that is called haploid, and that's what your gametes are, your eggs or your sperm. So those are haploid cells, and the shorthand for that is just N. So this unit, or this part of chapter 5 that we're dealing with now, is how do we start with a diploid cell, a body cell? It's just called an ovary cell if it's a woman, and if it's in a man, it's a testicular cell. But how do you start with a body cell that is 2N and produce cells that are haploid? So in this process of meiosis, what is going to take place is we're going to reduce the number of chromosomes that are inside a cell. So instead of having both members of a homologous pair, the cells will end up, your gamete cells, which are your reproductive cells, will have one member of a homologous pair. You'll have one number 1, one number 2, one number 3. Okay, so that's what meiosis is going to do. It's going to allow for us to reduce our amount of genetic information. And the reason why you have to reduce your genetic information is because if we didn't do that, the amount of genetics in each of our offspring would just keep doubling and doubling and doubling and doubling. So, most of your body cells are diploid. They have 2 copies of each chromosome, one from your mom, one from your dad. So now what we're going to do is work on how do we then reduce that number. So these are our gametes. They're haploid cells. Remember, your diploid cells are your body cells or somatic cells. So now we're either talking about somatic cells or we're talking about gametes. And today we're talking about gametes. They're haploid cells. So the process that does this is called meiosis. It produces cells that are haploid. It's very much like mitosis in that meiosis occurs in stages. So we have to take a person here who has 23 pairs of chromosomes and reduce it down to 23 chromosomes, no longer in pairs. And same with mom. Same thing's happening in mom as is happening in dad. And then when fertilization takes place, when those two join, the zygote that results, that first cell that results, will be back to having now its chromosomes coming in pairs. One from mom, one from dad. So here's a big, long, kind of little graphic example of what's taking place. And you can take a look at this. But I think what's important to notice here is that meiosis is a really specialized, very specialized process, and it's only happening in very specialized cells. So it's happening inside the testicles of men, and it's happening inside the ovaries of women. And it happens to produce these gametes, and then once gametes fertilize, from then on out, it is mitosis, 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 mitosis. So you get this zygote, this cell that now has the full complement, half from mom, half from dad, and it's just mitosis, mitosis, mitosis. And now all of these body cells are identical to that original cell. And as I was saying before, or perhaps I haven't said it already, but that is what makes the cells different from each other is not that they've got different genetic information, it's which of the genetic information are they using. Okay, so when it comes to talking about these chromosomes, we have just a little bit more terminology to get into. And so when we're looking at those chromosomes, remember they're coming in pairs. Here's the karyotype. One of these came from your mom, one came from your dad, one from your mom, one from your dad. There's no way for me to tell by simply looking at a karyotype which one of these came from your mom, which one came from your dad. But what it does allow for me to do is it allows for me to see the actual sets of chromosomes. And there are genetic conditions that are problems, and the problems are associated with not having the correct number of chromosomes. And so an amniocentesis is a process, a medical procedure that some patients go through in order to determine that the child has the correct number and the correct sets of chromosomes. So when you look at the chromosomes here in this karyotype, the first 22 of them are called autosomes. The last pair of chromosomes are the sex chromosomes. So the first 22 are autosomes, and they're determining, they've got the information all about your body on them. Auto means self, so it's about yourself. The last pair of chromosomes are called the sex chromosomes. They're the chromosomes that determine your gender. So females have 2 X chromosomes and males have an X and a Y. And if you take a look at the photograph here of these sex chromosomes, it's really obvious that this is not a homologous pair. They clearly are not the same size. They are not the same shape. And they clearly do not contain the same type of genetic information. So they are not technically a homologous pair. However, they will behave just like a homologous pair throughout this process. So you want to think of them as a homologous pair for right now, and only because they behave like a homologous pair, but not that they have the same confinements as a homologous pair. They are not the same size. They're not the same shape. And they do not contain the same type of genetic information. As a matter of fact, the X chromosome has lots of genes on it, hundreds of genes, and the Y chromosome has very few. And that might account for the size difference that you're seeing. So in this karyotype, this is a karyotype of a baby girl. And I can tell that because the 2 sex chromosomes are both X's in this example here. So here's what the chromosomes look like. And there are people who get jobs doing just this, working in labs, carrying out karyotypes. Okay, so the egg and the sperm, both of these are haploid cells, and when they fuse together in what's called fertilization, then you'll get back to it then becoming a diploid zygote cell. All right, so we're going to start working on meiosis, but I'm just about out of time for this first 15 minutes, so I'm going to come back and start the next video. We'll pick us up right here.